then you shouldn't have this kind of differences, but otherwise most of the cases you should have this kind of differences. These are called allotypes. That means we both are human being, but you are having different than me. And the third kind is called the idiotypic determinants. Now, what do you mean by idiotypic determinants? It is a type of determinants which is the only thing which is designated by the variable region of constant and light, both this case. So variable region. If you are having varying in the variable region, then we end up with different thing. So this variable region is varied from all the immunoglobulins that we are getting. So inside your body now, in the very beginning of isotypic determinants, we talk about a species and we found similarity in the members of species. For allotypic determinants, we found similarities among in my body. For example, we are having differences in your and my immunoglobulin, but if we take all of my immunoglobulin and check for allotypic determinants, all the allotypic determinants in my immunoglobulin are same. They will be same. But if we take idiotypic determinants, we will find different type of idiotypic determinants in all the different type of immunoglobulins that I am possessing, so or you are possessing. So you take only me or myself. Now you check all the immunoglobulins. One is so this is the M1 or immunoglobulin 1 for me, this is the second immunoglobulin for me. If we check both of them, they'll be having variations in their idiotypic determinants, because the idiotypic determinants are considered to be present in this FAB region or the variable region. As they present in the variable region, they will be varying inside the same organism, like inside me or inside you. If you take your immunoglobulins and compare them, you will have different type of idiotypic determinants, right? So these are the three different models of isotypic, allotypic and idiotypic determinants. Here are another mode of example. So you can see both this case we are having the variations in this FC portions, but we are having similarities in this uh, constant region of the light chain. So this is called immunoglobulins because we are having similarities in the whole species members, members of all the species. So all human beings are having this kind of similarity. Immunoglobulins of allotypes. Now if you look for allotypes, what we get, we are getting variations among you and me. So if you take immunoglobulin for you, it will be different than we take immunoglobulin for me. For the third kind, which is the idiotypic, what we get is that if we take the immunoglobulin of from my cell only, then all these immunoglobulins will be different inside my own body also. They are idiotypic, so they are much more versatile. So as we are growing from isotypic to allotypic, then finally allotypic to idiotypic, we are going toward much more specificity, right? Much more specificity. So this is the actual concept. Okay, this is the actual concept. So once we have talked about all these kind of basics of immunoglobulins, now let us talk about the five different types of immunoglobulins, because we need to talk about the structure of all these different types of immunoglobulins in detail. Now if you look at this picture is provided because just to tell you how to recognize one immunoglobulin. Now all this type of data that I've suggested you, I've told you about the basic structure and the basic functionality, and all these things are applicable to the immunoglobulin G or IgG, which is the most common immunoglobulin of all, which is found approximately above 70% in the concentration inside our body. So it's much more abundant, very much abundant, immunoglobulin, and they are having all this kind of structures. Now this immunoglobulin obviously is a monomer. Now if we try to remember, I want you to remember in this way, GAM D. So it's a GAM, GAM, then DE. So it's a GAM D to remember it. Why? Because this is the most abundant, so it should be fast. A is the second abundant, second. M is the third abundant, is fourth, then 4 and 5. Okay, so what we get, this IgG is a monomer, IgD is also monomer, IgE is also monomer, but these two things are completely different. By looking at their position, you can tell that this A is a dimer. Now usually this A is not a dimer. In this in normal form of A, it's also a monomer, but when it is secreted, it is forming this kind of dimer. And we are going to know that immunoglobulin A is having a feature of the secretion. Now in these different types of bodies fluids and secretion, we are going to find immunoglobulin A. For example, our tears and our different types of secretion, we are going to find them. And also sputum, tears, and all these cases. And immunoglobulin A is also pentamer. As you can see here, five different monomers are come